In Chapter 9, we're going to focus on protein synthesis and gene expression. But in the introduction to the chapter, it actually poses some questions about genetically modified organisms. So think about these questions for just a moment before we go into the actual content of the chapter. Would you want your children to drink milk or consume fruits and vegetables that have been produced using gene-altering technologies? What if your beloved dog died? Would you want to be able to clone him? If your grandmother had a debilitating disease, would you want her to be able to help treat the disease using cells obtained from human embryos? Do you think it is ethical to use animals as biological factories for the production of substances that are helpful in treating human diseases? Do you think human cloning should be embraced or outlawed? So consider these questions for a moment, and as we talk through the rest of Chapter 9, come back to these questions, and as you learn a little bit more about these things, we'll ask these again at the end and see if you have a different opinion of it once you know a little bit more. So genetic engineering, which is what produces these genetically modified organisms, actually began in the early 1980s. With the production of recombinant bovine growth hormone. So what they're able to do in the lab is to take genetically engineered bacteria, so to take bacterial cells, which are easy to grow and reproduce in the lab, they altered their DNA. So the DNA of the bacteria was manipulated so that its genes carried instructions for making bovine growth hormone. If you don't know what bovines are, it's cows, okay? And so what these growth hormones typically do in cows, which is what they're normally produced in, these growth hormones increase their body size, and in turn they increase their milk production for dairy cows. So an increase in this growth hormone would cause an increase in the production of dairy. And so what they did in the lab was they caused these bacterial cells to produce this hormone that's needed for growth, and then they take that hormone and they inject it in cows, increase their body size, increase their milk production. So what's interesting about this is not only is this used in cows, it's used in lots of other organisms that we'll talk about later on in the chapter, but it is a good illustration of the fact that we can produce any protein in lab or in a cell because proteins require the use of genetic information known as DNA, and we know that all living things possess DNA. So if you can change their sequence of genes, you can produce any protein that you might need. Now, in order to understand the process of protein synthesis, we first need to know a little bit more about DNA. Now, we've kind of pieced this information together, so this actually goes back to Chapter 2 in describing the basic structure of DNA, listing the groups of bases that make it up, and understanding what it means when we say complementary base pairing. So first off, we know that DNA stands for deoxyribonucleic acid. We know the job of DNA is to store genetic information, so it is your instructions on what to build and how to build. And we learned in Chapter 2 that DNA is made of nucleotides, so it consists of a sugar, a phosphate, and a base. The sugar is known as deoxyribose, which is where DNA gets its name from. We know that when the sugars and the phosphates and the bases are all put together, it forms a twisted ladder that we call a double helix. And if you look over to the side, you'll see our sugars and phosphates alternate to make the sides, and then the rungs of the ladder are made of nitrogen bases. Now, when we talk about nitrogen bases, there are 4 that are found in DNA: adenine, guanine, thymine, and cytosine. And these 4 nitrogen bases are put into 2 categories based on their chemical composition. So adenine and guanine are grouped together, and we call them the purines because they exist with double rings. That's how they're built. Thymine and cytosine are grouped together as in a category called pyrimidines, which are made of just single rings. So when DNA is made, your cells actually follow the rules of complementary base pairing, which says anytime you're forming DNA, you take a purine, and by using a hydrogen bond, you bind it with a pyrimidine. So A for adenine always binds with T for thymine, and G for guanine always binds with C for cytosine. And so because of the hydrogen bonds that exist and the rings that exist in each, you always have 3 rings that go across the middle. 2 for adenine, 1 for thymine. 2 for guanine, 1 for cytosine. So if you remember Auburn tigers in DNA, A goes with T. And if you remember George Clooney, G goes with C.